Okay, I'll start. Fine. Yes, good to go. Yeah. Okay. Good evening, everybody. So I'm Sharvari Kulkarni and I'm Technical Associate at BDG Lifesciences. First of all, I would like to welcome you with a great pleasure to see your enthusiasm and willingness to learn this basic course on bioinformatics. Okay, in which we will be covering basic tools and databases and the introduction to the subject. Okay, so first I will start with my brief introduction, then I will talk a little bit about the company, and then we'll move ahead with the actual topic. Okay, so I'm Sharvari Kulkarni and I'm working as a technical associate for BDG Lifesciences. I have 10 years of experience in the field of biotechnology and bioinformatics. I was working as an assistant professor for biotech engineering, and my area of specialization includes bioinformatics 1 and 2 and uh, IPR, bioethics, biosafety, and bioinformatics, immunology, and immunotechnology. So today we are going to focus on bioinformatics. I believe you participants, you all must be from the background of life sciences, but you might not have come across knowledge about bioinformatics, or you might be having a little bit idea about it. I will try to make this 2 hours productive to you so that you understand at least central dogma of bioinformatics. This is the basics and 2 hour session. So we'll be focusing on the introduction to the tools and databases, and we have our separate module or workshop on it for 10 days, in which we can include hands-on training on all these softwares. So if you find this webinar useful, and if you have interest to learn and take hands-on training on it, we would like to invite you for the 10 days program, which is combination of basics and advanced. Okay, so let me share my screen now. Meenakshi, are you there? Can you tell me if you can see my screen? Please unmute yourself and tell me.
Yes.
Is it visible?
It is showing like like fine art for gateway.
Okay, but the screen is shared, right? Now I'll show you my PPT. Tell me if it is
Ma'am,
visible.
it is
It's
shared.
visible? Yeah, fine. Yes, so we'll start with so bioinformatics, a new frontier on life sciences horizon. Yeah. So let us begin. As I've given my introduction, I would like to begin with company's introduction in short. We are BDG Life Sciences, our group, biodiscovery group of life sciences. Our group was created in 2009 for students, researchers, scientists, professionals, and who are related to the field of biotech, bioinformatics, and pharma. Okay, so we have free consultation, tutorials, books, projects, reports, assignments, and training job offers in India and abroad. about the company. We have around from 2010, we have our wide array of services, which includes research projects, workshops online and on-site, training online and on-site, webinars, internships, contract research, seminars, and conferences. These are, in short, I would like to enlist some services which are offered by us. As I told you, workshops on bioinformatics, basics and advanced, structural bioinformatics, CRISPR, dynamics and simulation, immunoinformatics. Then we have docking workshops. Then we have R programming and cancer data analysis. Now let us move ahead with the topics. So let us start with what is bioinformatics. Basically, what is the need of bioinformatics? If you all can see in this slide, it starts from 2000 that the huge generation of biological data started. Now, because of the availability of this high amount of biological data, of course, generation of data is not just important. We have to actually organize, we have to first store this in the form or in proper form, and we have to utilize or organize this data in a proper way in order to analyze and interpret the result. So we can see from 2000, it started giving us huge amount of data. We are here now. Until 2025, it is expected to give lot of data in around, I can say it's predicted that data will be as high as 2 to 40 gigabytes. So now some science or technology has to be there in order to handle this huge amount of data, and that is where bioinformatics come into picture. So I'll start with some basic simple definitions of bioinformatics. It is the computational branch of molecular biology. It is multidisciplinary branch with vast applications. Why I'm calling it as multidisciplinary is because of its you can say logic, that it is taking help from different disciplines, as well as more than that, it is helping many disciplines. So we are going to call it as multidisciplinary branch. Okay. Now, again, moving ahead, I would like to define this bioinformatics as conceptualizing biology in terms of molecules and then applying informatics techniques to organize the information associated with these molecules. Okay. So basically means what? Concepts are used which are in basic biology, and then we are applying information technology, so that there are development of some information technology based programs and which associate with the molecules on large scale. Now, how does the workflow go? Is it starts from the sequence, then 3D structure, and then function. Okay. Yeah. Now we have, I would like to depict this bioinformatics in terms of one picture. So this is a living cell, and we are making perturbation. Perturbation means what? Disturbance to living cell. And that is how it gives dynamic response. So it is not a static system. Living cell, though it appears to be static system, it is not. So whenever you are giving any disturbance to living cell, it will be it will give you dynamic response. Now, what are we doing exactly in bioinformatics? We are converting this living cell to virtual cell, right? So virtual meaning what? Something which is not existing, but with the help of but with the help of this, what you can say, with the help of this softwares, we, just a moment. Yeah. So with the help of software and technology, we are creating a virtual cell. That means we are going to make a living cell in computer such that it exactly mimics the behavior of virtual living cell. Fine. So now this virtual cell is made with the help of, or its replica of living cell is created with the help of bioinformatics. And what type of knowledge is given? It's a biological knowledge which is computerized in the form of, this information can be in the form of sequence or in the form of structure. Okay, so we can provide this biological information in the form of sequence or structure, and then it will be utilized to virtual cell, and then we are going to apply some principles and come to the practical applications. So that is how these practical applications, again, it will help the, what you can say, uh, wet lab in order to give some analysis. So that is how wet lab and dry lab is connected to each other. Now, the highest step of this is mathematical modeling. So you build mathematical models based on this. Little bit of theory. Term bioinformatics was invented by Pauline Hopfok and Ben Hasper in 1970 as the study of informatics process in biotic systems. Okay, Pauline Hogwarts is a Dutch theoretical biologist and complex system researcher. Now, from where it actually started? It started with Human Genome Project, which initiated in 1988, and it declared complete in 2003. So, which were the major goals behind this Human Genome Project? It was to determine about 3 to 10 to 9 base pairs of and identify 50,000 genes. Okay? So, it involves lot of computational tasks like storage and indexing, building context, and scanning for genes. So if you see here graphically in the form of bar diagram, it started, as I told you, in 1988, and you can see its information is getting increased, and till 2003 it got completed. So this is the finished part and this is the unfinished. Now some milestones we are going to discuss in bioinformatics. So basically started with Watson and DNA model in 1955. Then some important milestones only I will highlight. PDB was discovered in 1975, which is very important milestone. Till the date also we use PDB. It's all possible because of protein data bank, that all the softwares are connected and all the files which we are going to use are necessarily PDB files. Then we have our NCBR, which was developed, which is also important database, which is like consortium. So I'm going to teach you how to operate this NCBR and PDB. Then we have some important algorithms like BLAST algorithm. Then we have SwissProt also, which we are going to discuss. So three important databases: NCBR, SwissProt, and NCBR, SwissProt, and PDB that we are going to discuss. So in short, this is all covering what is bioinformatics and how it is called as, why it is called as multidisciplinary. So here you can see it is giving application of DNA sequence analysis like BLAST, clusters, etc. Then it is MD simulations where simulation means you're going to simulate the system. That is also, these all are itself different module, or it consists of our one workshop. I'm going to highlight here, and we actually conduct different workshops on different topics. Like simulation, you have to learn, which is programming based, and we use SNAP or NMD for that. And then you have drug designing, in which there are steps. Today's pandemics, drug designing and discovery has got a lot of importance because it involves target identification, target validation, lead identification, lead optimization, etc. Then we have analysis of, so this all like you're getting it. Bioinformatics is a central thing and it all goes on different lines of branches. Simulation is one application, drug designing is one application, protein sequence analysis is one application, like in finding out the properties like amino acid composition, PR, domain analysis, secondary structure analysis, etc. Then again, one stream goes to phylogenetic analysis. Then one stream goes to molecular interactions like docking, then protein DNA interactions. Then you have molecular modeling, in which function prediction and so now let us start with what is data. Databases are basically libraries of life sciences information collected from scientific published literature of high throughput experiment technology and computational analysis. So what is the source? That database. Any database in general means what? Something which stores the information. Okay. So basically library of life sciences means the database is nothing but it is a library of information which is based on scientific experiments. So source of information is scientific experiment. And they are broadly classified as sequence database and structured database. Okay, so they are also classified as primary and secondary. Primary means like NCBI and all, the main consortium. Secondary means developed. Okay, so and along with that, based on the purpose or based on the criteria of what type of information is stored, it is sequence database and it is structural database. Okay. Sequence databases stores information about the sequences and structural databases stores the information about structure. So these are some highlights. There are many databases. So when we started, there were only three databases. But now we have around 1500 databases with us. Okay? So this is DDBJ. These are some important databases and we are going to learn these three databases mainly. NCBR, CRISPR and PDG. Okay, so this is NCBR. Now this NCBR is the home page of NCBR. Here you have drop down menu where you can select the databases. So in the workshop we actually take this as one edition I perform, which students perform another edition. So you have selection of databases here. So if you are interested to all databases, you can keep it as else you can select protein or nucleotide like that. And in the search, you will simply type whatever is your query. For example, here I have typed gene and here I have typed supplement. It will take me to this page where I have all information about using one go. Okay? So like application number, then it's everything about it. And most important is faster sequence. So you see here, there is one option there. Now it is very important before proceeding in any bioinformatics based software tool to know what is because faster sequence starts always with greater than symbol. So you see here, starts with greater than symbol, then accession number, then the species name and the actual sequence. Very important. Copy and paste. Wherever we are using any tool, we are going to copy and paste the password. So unless and until it is correct, tool is not giving proper result. So it always starts as a tool. Even if you use this single greater than symbol or any space, it is not going to give a correct. Remember, whenever you are operating any tool, you have to correctly copy paste this. So this was about the NCBI. You can also explore it for yourself. If you click on various items, you will get lot of information. Okay? Then we come to PDB, which is Protein Data Bank. This is the home page of PDB, and where have very important information, that is 3D structure coordinates of protein. So those are very important. But point of view of getting any analysis like docking, simulation, always tool will ask for PDB file. Okay? So we have to use the PDB file for any and everything. So it is important to learn what is PDB. PDB is data bank, and here also you are going to simply enter the search. So if here I'm using the start type glycoprotein, I will simply enter here the search term and go on search. So they give me this particular entry. IUWRG is known as PDB code. Remember, always PDB code is made up of four letters. First is most of the time is a numerical, and next three are alphabet. So here we go to the right, and here we click on PDB format. Now this is the format. We have to get this plain PDB format, which is not PDB file. So whenever you are downloading, the file will be downloaded at iuwrg.pdb, and you don't have to take all this format. These are all text format, and we are interested in format. So this is how you download PDB file. It will be installed or downloaded automatically. The next part is unit. Next part is unit plot. As I told you, it is also very important. Unit plot and stress plot. So here also in the unit plot you are going to notice why unit plot, because it is manually created and annotated by scientists. So unlike NCBR, from unit plot you will get the sequence of the protein, faster sequence of the protein. From NCBR also you have to get, and you will get lot of results from one protein. Like in uniprot, you get quickly the result. And for one protein, there is only one. Okay, so if your protein is here, you type your search, you get results in more quickly and more. For example, here I use SARS-CoV-2 protein. I will take me to this page where gene function is given. Also, you have options text and pasta canonical. So you click on pasta canonical and it will download the pasta. Next, this is about the database. Now I'm going to highlight everything and everything. If time permits, I'll show you demo of one of the databases. Now here we come to primary structure analysis. Primary structure analysis. First step should be always to know the properties of the protein. So now we know the sequence by database. Now we move ahead to the so property meaning physico-chemical properties. And there is an excellent tool which is known as prod scale, which is operated in an expressive server. So in the prod scale, you put accession number, and then on the basis of accession number again, uniprot accession number, as I told you. So now we will be able to retrieve uniprot help of accession number. So always uniprot accession number is start EO and four letter code. Yes, please. Now I'm audible. Somebody from the participants, please reply. Meenakshi, am I audible?
Yes, ma'am.
Okay, fine. Okay, so now in the prod skill, we are going to put accession number, and here you have this property like molecular weight, bulkiness, polarity. You can select any property and then here by default hydrophobicity is selected at site and do. Okay, you can select any property of your choice. Since it is by default, why? It's because most of the times interest of researchers is to know the hydrophobicity. So hydrophobic regions are detected by means of this software of Crossfield. And you can see you must be wondering why hydrophobicity has too many options. Okay, it's because they are after the research paper of the scientist. So title level is moved. Now you paste your sequence here and then click on hydrophobicity title. Then you will get this kind of analysis. This is the fraud scale output. The beautiful graph you get for amino sequence. Now on the x-axis you have this amino acid positions and on the y-axis you have score. Okay, so this is like how to read this graph is after the amino acid. It's always our x-axis gives the amino acid position and y-axis gives the score. So on the x-axis are how to read this graph is highest peak is red. Okay, you can see here the highest or tallest peak is at around 10th position. You can see that 10th amino acid is the region where hydro. After that you have this 40. After that you have this around you can say 80. So like that peaks and troughs are red, and on the basis of peaks and troughs you have just a moment. Hello? Yeah, yeah. Okay, I'll just shut off my Bluetooth and take the normal speaker. I'll check. Yeah. Okay, wait a minute. So, how to get a response from yourself? Is there any problem in audibility? I'm getting a complaint that it is not clearly audible. Now, is it clear?
Yes, ma'am.
So, should I repeat from some point where I was not audible? Do you want me to repeat? From where should I repeat?
Yes, ma'am. From
PDB?
data.
PDB database?
Yes, ma'am.
Okay, I'll repeat
Yes,
it from
ma'am.
there. Yeah, fine. I'll repeat it from there. And even
Okay.
if in between it's not clear, then please feel free to unmute and tell me. Otherwise, I will not understand. Okay?
Okay. Okay.
Fine. So I'll come back to PDB and talk to you again. So all the prod skill part you missed, that means?
Yes, ma'am. Like your voice is not audible in that time.
Okay, but it is now clear, right? So I should go ahead with this. Hmm.
Yes, ma'am.
Yeah. So I'll come back to PDB and then we will talk. Yeah, so now PDB. Basically, PDB is a protein data bank in which we have our PDB files which are stored. Okay, so PDB file means what? It is 3D structure coordinates. It saves 3D structure coordinates, and this is very, very important. Wherever we are operating any tool in bioinformatics, you have to get the PDB file. So always PDB file format is saved and used. Fine. So here, how to download PDB and how to operate PDB, that I'll show to you. Yeah. So here we will simply enter the search term. So always remember PDB code starts with, here I have used same example like SARS-CoV spike glycoprotein. Okay. Now, here I will type in the search box, I'll type SARS-CoV-2 protein and it will take me to this entry. And this 5WRG is called as PDB code. Always remember it starts with numerical number and followed by capital letters. So in this page, you will get all the details about the protein. 5WRG is the spike glycoprotein. And here you can see there is option of download files. So how to download PDB file is the first most important thing in operating all the tools. So you have to go on download files and then PDB format, GZ. So you will click on PDB format and not all these formats. So you will strictly get this PDB format, which is a coordinate format. And we are interested in coordinates, not in the text. Other are text files. So automatically when you click on PDB format, it will start downloading in your system. So without any pain, it will be saved in your system. So you have to just save it and use it in the software, whichever you want. Then we talk about UniProt. Why UniProt if the sequence is obtained from NCBI is the question which is asked many times. NCBI is giving you sequence, but it is a consortium. So it deals with lot of information. So you have to filter the sequence for a single sequence of the protein. You have many entries. So instead of that, in UniProt, you have facility to get only one entry per protein. So that becomes simple for us. It saves time. Also, it gives you brief description as well as brief description of structure and function. So you get concise information in short time. So this is how UniProt looks. Again, I told you, we get the accession number if you know. If you don't know the accession number, then you can put the description. So it will take us to this page where you have function and you can see here, view the format. It's FASTA canonical. Okay. So in the FASTA format, you take the FASTA sequence and you can download it. Okay. As we did it in NCBI. Then we come to data analysis. Data analysis basically primary structure analysis, secondary structure analysis, tertiary structure analysis, function prediction, and sequence analysis. Okay. So first we focus on primary structure analysis. Now we know the sequence. Now we know how to derive sequence file. Now we know how to derive structure file. So now we move ahead one step, and that is primary structure analysis. See here, we are using Prot scale. Prot scale is basically a tool which gives properties of the protein, physicochemical properties of the protein. So it gives us a profile which is produced by amino acid scale on the particular protein. So what is amino acid scale? It is basically the value which is given for each type of amino acid for each particular property. That means hydrophobicity. You can see here, these are the enlisted properties. Like 50 predetermined scale values are entered from the literature. So we have this a much amount of information, and we are going to use this information and predict the property of the protein in particular region. So can you see here, all these options are there, and here now we learn how to get this uniprot sequence, or else we have also learned, if you have uniprot accession number, then we can put uniprot accession number. If we don't know uniprot accession number, then we can paste the sequence in this window. Okay, now we've select, you can see here, this hydrophobicity kite and doolittle is uh, actually this hydrophobicity kite and doolittle. It is by default. Why it is shown? It's because most of the times it is the property which is, you can say, uh, studied by researchers in order to come to some important conclusions. Like if hydrophobicity is more in that particular region, what will happen? It is a transmembrane protein. It is likely that it functions as a receptor. So we have this property. Now we can use copy paste the sequence. Very simple to operate. You have to just click on the submit button. Then it gives us this beautiful result. Okay. So you have this peaks and troughs. Wherever there are peaks, you can extrapolate it on the x-axis and see which is the region with that highest property. Now, if you see here, highest peak is at around 10 position. Then we have 40th position. Then we have 80th position. Then we have 100th position. So all these peaks are, you can say, all these peaks are noted, highlighted, and then we talk about its predictability. Like here, hydrophobicity, I can say, is at 10th, 40th, 60th, no, not the 60th one. You can say this 70 and then 100. And drop down is at around 55. So that is the least hydrophobic region. That means it will be hydrophilic. Yeah, then next part is structure visualization. So this was the primary structure analysis. Then we come to the structure visualization, how to see the structure of the protein and alignment by a very interesting and user-friendly tool, which is known as Swiss PDB viewer. It is basically a comparison of two proteins. Can you see here? So this Swiss PDB viewer is not online tool, but it can be installed online. So you go to Google, simply get the Swiss PDB viewer and get it installed in your system. So it is locally installed. If the correct version of Swiss PDB viewer is installed, then it should give you this 3 pop-ups window in which this will be our platform of working. So we will load from here file. You will get one option to open PDB file. So we will open our PDB file, then it will show like this beautiful depiction is obtained. Here you have another applications also like display, build. It is also used as editor. Sometimes when you're using PDB file for docking and simulation, you can add the residues. If you're interested to work on many specific residues, then you can delete residues. So Swiss PDB viewer basically has a lot of applications with respect to PDB files. Now, most important application when you are comparing any two proteins. So we take an exercise of comparison of like two hemoglobin molecules. I usually teach this with the help of hemoglobin molecule. So we compare two different hemoglobins from two different species. Family is same, so they will have definitely some sequence and structural similarity. So if the structural and sequence similarity is there, that means it will align on one another very nicely. So if you take these two structures, can you see here in the window, there is one option of window control panel in which you give different colors to protein, and this is how they superimposed on one another. And in the alignment window, you get something which is known as, can you see here? This is little small. When you are actually performing, you can increase the font and see, okay? So you get this superimposition. You can see this heme white is getting superimposed on one another. And then you have this RMS difference, okay? So RMS difference is basically root mean square deviation. And what is root mean square deviation? Always remember, this is very, very important terminology in bioinformatics. Root mean square deviation is basically a deviation from the reference structure and the structure which you are comparing. So between two structures, we are comparing one atom of reference structure to another atom of the template structure. So one structure and second structure. If I'm talking about hemoglobin 1 and hemoglobin 2, comparison of first atom of hemoglobin 1 and comparison of first atom of hemoglobin 2. That value is seen here. Now, if that cutoff value acceptance is 4.8, so if the value between two amino acids is below 4.8, then only it is acceptable, like 3, 2, etc. If those values fall above the 4.8 beyond the scale, then we can say that these values are not acceptable. So we discard that kind of alignment and we say that it is not a significant alignment. So this is also one of the very important and interesting interpretation. Now, if you club whatever I'm saying, you can say that step by step we are getting information about the protein, which is not known to us. So this is kind of one whole project of in silico analysis of one protein. Okay. If you just know the sequence, how you can reach to different applications and get all the information about the protein just because we have developed these tools and databases. Next is RASMOL. RASMOL is also structure visualization, which is very simple as compared to Swiss PDB, where you have to again, what you can say, locally install this RASMOL. So it will be installed in your system as RASWin. Then comes secondary structure prediction. So this was about the primary structure analysis and structure visualization. Then comes another important part, that is secondary structure prediction, in which we discuss Chow-Fussman algorithm, very famous and interesting algorithm to run. It is sliding window technique. So see here, Chow-Fussman is very simple to operate. Like this is the server, you have to just put the sequence and you'll get this interpretation. But as a bioinformatician, you should know what is the algorithm. So always try to understand the steps of algorithm. Though it is painless, tool is going to do everything for you, but it is not just important to get the result, but it is important to know the algorithm, how you're getting the results, and to analyze the results. It's very, very important. So sliding window technique, what do I mean by sliding window? I'll tell you. P alpha, it is 4 out of 6 contagious. P beta, it is 3 out of 5 contagious. Okay, so what do I mean by this sliding window technique? There are this P alpha, P beta, P turn values, which are assigned according to the propensity of the amino acid. That means there is a tendency, specific tendency of each amino acid to be present in specific type of secondary structure. Like P alpha tendency is one value, P beta there is one particular value. So depending upon these values, sliding window technique is run. So for example, if I'm saying here, P alpha is 4 out of 6 contagious. That means at the time I'm considering 6 contagious residues, and 4 out of these 6 contagious residues, if they show P alpha greater than 1, the probability of alpha presence is greater than 1, then that particular region is assigned as helix. Now you move ahead one amino acid at a time. So one amino acid I'm shifting in the sequence. Then I will scan again out of 6 residues if 4 are giving me more than 1 value. Then again, I will shift by one amino acid and I will check if out of 6, 4 contagious amino acids are getting me value of greater than 1. So like that, you keep on scanning for alpha helical regions first. And then you keep on scanning for beta sheets in similar way. Like out of 5, if 3 are giving you more P beta greater than 1, then declare that region as P beta. Again, shift by one amino acid. See if P beta is greater than 1 for 3 amino acids out of 5. Okay. So like that, you will declare that region as P beta. Now, whenever you're getting overlapping regions of P alpha and P beta, that time, of course, the dominance, if P alpha is greater than P beta, then decide that region as alpha helical. If P beta is greater than P alpha, then decide that region as P beta or beta sheets. Rest of the region is declared as turn, but nowadays we are also verifying it with the formula of Fourier series like f of series, where we consider the middle residue as j residue, and like this f of j plus 1, f of j plus 2, and f of j plus 3 is calculated. Now this is the server actually, where we are going to again, as see here, always it is mentioned that you have to submit the sequence in FASTA format. So now what you can do is, if I tell you to get Chow-Fussman result or secondary structure prediction for suppose oxytocin, then we'll get FASTA sequence of oxytocin and paste it here, then go for predict. After predict, clicking on predict, it will give us this prediction, which is very simple to read and accurate. Okay. So see here, it is read in this pattern: helix, turns, and sheets. So wherever green symbols are there, you can see it is EEE, that means sheets. Wherever HHH symbol is there, it is helices. And wherever there is T symbol, it is turns. And rest of all are C, that is coils is also there. This is rare, but sometimes you have some coils in the structure. And best part of this is at the end, you have total number of residues present in helices, a beta sheets, and this turns. So it becomes easy and perfect for you to understand in which region helices, sheets, and turns are present. So all this, if you understand, just if I give you some advice to note it down, and then you can explore or for yourself how to operate it, or the best ways you can get our workshop. You can join our workshop where we perform these exercises. Next part is sequence alignment. This is again very, very important thing to learn. Aligning two sequences gives us information about structural, functional, and evolutionary relationship. So sequence alignment is you align two sequences in order to decide what is the similarity between two proteins. And if there is similarity between two proteins, of course, they are structurally, functionally, and evolutionary related to each other. Now, there are two ways of alignment: global alignment, local alignment. Global means entire length of two sequences is analyzed, and local meaning short stretches of the sequences are aligned. So in practice, what we prefer is local, because we deal with large number of residues at a time. We have large number of residues in one protein, not the small proteins which we are dealing with. So we go for local sequence alignment, and BLAST that we are going to learn is a local alignment search tool. Now, on the basis of how many sequences we are dealing at a time, there are two possible ways of alignment, like pairwise sequence alignment and multiple sequence alignment. So in pairwise sequence alignment, at a time, two sequences are studied. And in multiple sequence alignment, at a time, more than two sequences are studied. Okay. So I've taken this simple example for you to understand. We are comparing ATCG and TCA. If you compare these two sequences, you can see here ATCG and TCA. If they are aligned to one another, you can see here I put gap at the first place, and there are three residues. So TT is a similarity, CC is a similarity, and GEA is a mismatch. So this is a match, this is a match, and this is a mismatch. So what would be the score? Two matches. If you see here, there can be more than one ways to align, right? One person can align it this way, another person can align it this way. This can be another way where we put the gap at the end, okay? So ATCG aligned to TCA. Now let us see how many matches and mismatches we are getting. It is mismatch, it is mismatch, it is mismatch. So there is no single match, but here in this case, there are two matches. So our purpose is always to align the sequences in such a way that there are more number of matches and less number of mismatches. Okay, so what would I say? This is the good alignment. Now, this is something like a production of gap. Now, I'll give you one day-to-day example for this. If you are giving one competitive exam, there always a match or correct answer will give you positive score. So, if I decide to give positive 2, plus 2 will be for a correct answer. If there is a mismatch or there is wrong answer, it will be 0. And if you don't attempt the question, it will be -2, right? So gap production in case of alignment is like an attempting, or is like not attempting the question paper or question, okay? Yeah. Now, this is how one important thing to understand is scoring scheme. So if you compare these two sequences, you can see here, uh, this is the scoring scheme. -2, A, and there is a gap, so -2. T and T, so it is +2. C and C, it is +2. G and A is -1. The score is -1. So you calculate the best score here. If you align, this is the best score, right? So why? Because +2, there are two matches. This is a mismatch and this is a gap. Now, if you talk, these two sequences have taken two different sequences, ATCG and TGGA. So it is distantly related, right? So we don't consider this kind of alignment. Whatever possible ways you try to align these two sequences, they are distantly related. So tool will not give you the positive results for this. So it will be giving you least score, and which is not acceptable. So the alignment is not significant. Now, this is the blast. Here in the blast, we can give, first we will go to protein blast. So if you are performing comparing to, there can be two possibilities, either a nucleotide blast or a protein blast. So protein blast is always a comparison of two proteins, okay? And nucleotide blast is comparison of two nucleotides. And there are again different flavors of the blast, which are depending upon if you're using the nucleotide blast and comparing with the protein sequence, then there is T blast N. So depending upon that, uh, we go for a blast, okay? So now I'm going to tell you about the protein blast. So if here you can enter the accession number. This is a blast home page where you have to put some information. So here is the accession number 01295. I have taken one example. It is a gamma fibrinogen protein. So automatically your job title will come, and then you have to choose the database. Now I'm comparing a protein, so I select UniProt database, which has protein sequences stored in it. Then after that, I can keep everything as default. You can select the organism, or you can limit the organism if you want. Then you can click on blast P and you run the blast. Now this is the alignment. Here you can select the, this is how you get the result. Isn't it fantastic? So automatically you get the highest hit at the top. So you can say that if I have entered gamma fibrinogen of one species, the most similar protein to my query protein is Bos taurus fibrinogen, then Homo sapiens, then Rattus. Okay, so this gives us along with the maximum score. What are the scores of the alignments? Those are given. Always at the top, you have those entries which have maximum score along with the E value. Okay, so E value has to be less and score has to be maximum. So those hits are selected which gives you maximum score and least E value. What is E value? E value is the expectation or error value. It is the probability of occurrence of your results without error. So lower the E value, your results will be significant. Whenever you are reading blast results, you always go for those values which give you good score and less E value. Now, if you have this options graphic summary, in which you can see it simply by means of graphics, which is very easy to read like this. So top hits will be always like a bar diagram. It is shown. Pink bars are always scores which are less. You have this score and color codes also. So 80 to 200 is the maximum score. You can verify it with the blast results, but most of the times you get results which are greater than 200. So you get beautiful red bars, but unfortunately in this protein you don't have anything which is giving you more score than 200. So we get all the pink bars. So green, blue and black are most of the times not considered. So only those bars which are giving you score till the pink range, that means 80 to 200 are selected. Okay. Now you have next option of alignment also. You can see the alignment 1-to-1 and you can see its lineage also. Fine. So this is how you operate. Now, whenever you are dealing in practice, why we do it? You compare and get the analysis like this. First three hits to your protein. So that means these two proteins are highly similar to your protein. Okay. And see here, these are the accession numbers also which are given. So we can directly go to PDB. Yes. Okay. Now let us move to the next section, and that is primer designing. This is also very, very important thing to understand. Primer designing is you can design a primer for PCRs and various important applications using NCBI tool, NCBI Primer Blast. How to use this tool is simply type gene and name of the gene, that is albumin Homo sapiens. Now, why it is important to design primer is because you can start the PCR. You know what is PCR and primer, right? So primer is the short stretch of sequence from where there are forward and reverse primers. So short stretch of sequences from where you can start your sequence synthesis, right? So I have taken here an example of Homo sapiens albumin, in which you enter, you remember I had told you about NCBI drop down menu. There in the option of drop down, we have gene option. There I have shown you one example of protein. Similar way, you have to select gene and Homo sapiens albumin. Yes, so it will take you to the genomic sequence, complete genomic sequence of Homo albumin. Okay, now in that we can have, if I click there, I can see here there are about 15 exons. Exons are the coding sequences. Now, since I want to design a primer, what I can do is I will select one exon only and zoom in. So I'll get this kind of view. There are 15, you can count 15 exons. Like this, I can focus at a time on one exon. So now if I focus on this exon, what will happen? You can select a region behind it and forward it to design the primer. So how to do it with the help of tool? I'll show you. See here. Here is option manually. You have to just go on this portion and drag it back and drag it forward. Fine. So this will select your region. Always remember one tip. Leave 10 to 20 nucleotides up and down and drag this mouse over some region which is not very big, which is not very small. Once you select the region, give a right click and there will be option to blast and primer search. So if you click on this primer blast selection, it will in a short stretch of time, you have to click on get primers and then you'll get this primer analysis. Okay, so this is very important application again. In a single go, you get for that particular exon 10 pairs of forward and reverse primers. So you have primer 1, forward and reverse. You have primer 2, forward and reverse. Like that, you have 10 primers. Right? Now you can select this primer sequence. You get detailed primer reports also. So I can say for that particular exon, this can be my pair 1, pair 2. On the basis of this, you can select the primer and you can run the PCR. So this primer blast technique is used extensively in diagnostic purposes. Like if you want to detect the presence of particular type of infection, particular type of DNA in your sample, you can design a primer with the help of this and getting the sequence which is available in your gene database, GenBank or NCBR, design a primer and check for the detection. Okay, so it has got a lot of importance. Next, we are going to talk about, now we have done pairwise sequence alignment. We have also used primer designing, primer blast. Now the next step is MSA and phylogram. Multiple sequence alignment and phylogram. What is multiple sequence alignment? Multiple sequence alignment is when you consider more than 2 sequences. So like we performed blast, we performed it for 2 sequences, only the extension of it is multiple sequence alignment. So at a time, you can consider more than 2 sequences for about 15 to 20 at a time. Now, on the basis of this multiple sequence alignment, we use Cluster. There are different tools for getting multiple sequence alignment. Cluster W is the first one which is introduced. Then Cluster Omega is the modification of Cluster W. And now we have also Mega X, which is also an excellent tool for this analysis of sequences, multiple sequence alignment. So we first do multiple sequence alignment and going ahead on the same line, best application is phylogram. You construct phylogenetic tree on the basis of these sequences which are selected on the basis of multiple sequence alignment, and you can in detail talk about the similarity of the protein to another proteins, also about its homology. That means how if they belong to common ancestor and where in taxonomy, where in evolution exactly what that protein is present. Okay, so that is also one important application. Whenever we are talking in phylogram terms, we always have these terminologies which should be known to you. Root, node, clades. So root is basically from where the branches start. It starts from where the analysis starts, is the root. Node is the point where the branching starts. That means if these particular proteins are present at common point, but from there they are diverging. This is called as node. Clades are the groups. If they are present, if they show similarity, they belong to one group. So we'll take one example for this. Yeah, so this is where I have taken gamma fibrinogen protein in 7 different species. So I will go to Cluster Omega through Google, and then I will get this page of multiple sequence alignment in which you have to paste your entries. So I put all the numbers. What you can do best is you can go to NCBI, you can go to Swiss Prot. Then at a stage, if I'm considering 7, let us construct ourselves to 3 as an example. So I'm taking 3 different proteins. I will get their uniprot sequences and copy paste their FASTA sequences in this window. Once you copy paste this FASTA sequences in the window, you submit and get this kind of result, beautiful result you get. See here, I've taken only 3 proteins. So you can show it with the help of colors also. Wherever there are, you can see bright red color is there and star symbol is there. That means that particular residue is conserved throughout. And in between, can you see here, gap is there. So this particular, can you see here, SLL. So S is substituted for L at once. So there is gap. R is conserved throughout. So star. L is conserved throughout. So star. L is conserved throughout, so star like that. And one dot meaning substitution for ones, and a gap, two dots means slight conservation, that means substitution for two times. And gap is not at all related. So that way depiction is given. So by simply watching at colors also you understand about the similarity. Also, you can construct a phylogram. So this is the phylogram. You select on phylogenetic tree or guide tree. It gives you the best way of depiction. Can you see here? Yeah, these three sequences are aligned, are put on the phylogenetic tree like this. How to read this tree? I'll tell you. This is what I mean by root. So from there, it starts is the root, and from here it is branching. So this is first species, this is second, and this is third. Now you see, first is chicken, second is human, and third is panther. So I can read this tree as how to interpret this tree is very, very important. So these two are related, they are present on the one node. So that means human protein and panther protein, they are highly similar to one another, and this point is called as node from where the branches start. So they belong to common node, one node. Also they are showing equal branch lengths. So because they show equal branch length, that means they are highly similar to one another. And clade meaning this, where the two proteins are similar, they are present in one group, you can say, then it is said that it is present on one plate. And you can see here chicken is present on separate branch than these two. So we can interpret this in such a way that chicken is the protein, gamma protein or of chicken is related to gamma fibrinogen of human and panther, but at a larger distance. But they belong to common root, they don't belong to common node. Whereas these two, human and panther, they belong to common clade. Also, this branch length is similar, so they belong to common ancestor. Okay, so this is cluster omega. You have also facility here. You can see cladogram and real branch length is selected. So the branch length here, the guide tree is just the depiction, or phylogenetic tree is just depiction. There is also a way to see this with the help of scale values. Is there branch length corresponds to the length of the time of evolution. Now the next portion that we are going to talk about is interpro scan. Interpro, so by far we have discussed about structure, right? So we have discussed how to get the files, we discussed structure visualization, we discussed secondary structure prediction. Now we are going to talk about function prediction. So how to predict the function of the tool. This is also one of the very, very important and excellent tool, InterProScan. It is also like a consortium. So it takes you to the various databases from one database or single search. So since it is redirections to many databases, at the time you get a lot of information about the families, domains, and on the basis of domain analysis, function prediction. So what is domain? Domain is basically a particular, you can say, stretch of sequences or block of the protein, which is a repeated pattern. So that particular sequence or that particular block is associated with particular function. So it keeps on repeating in all the species and it is responsible for particular function. For example, glycine, alanine, alanine. So this particular patch is supposed responsible for one type of activity, like you can say it is related to the function of blood coagulation. So that particular pattern will be repeated in all the species and it will be always assigned with blood coagulation. So this is how we filter out similar domains in proteins, and on the basis of that, we predict the function of the protein. Yes, so here InterPro Scan is also online tool. So you know not installed. So by far you must have observed we have installed only two tools which are locally installed. Swiss PDB Viewer and Resmol. Rest of the tools are locally, online use. So this is the page of InterProScan in which you paste the sequence. Here you have facility to paste the sequence, but again, you can see here it is in the FASTA format. Once you select copy and paste the FASTA format, you can go for search. Okay, then you search the Interpro and you get these kind of bars. Okay, so you can see here there are 1, 2, 3, 4, 5, 6, 7, 8 bars. So what does this bar indicate? First bar, green bar, is the Interpro entry. So best part of this is wherever you move onto the mouse, it will give you the information about what the information it consists of. That is the best part. So even if you are very naive and you don't have any information, you just have to hover your mouse on each and everything and it will redirect you to that information. So it is the Interpro entry. Then it takes you to the, if you click here, it takes you to the Panther entry. Panther is the database for gene annotation. So all the gene information you will get in the Panther database. Then you have Smart database, which is related for domain architecture. Smart modular architecture tool. That means it is give you on the single click, it will take you to the Smart database over here and it will give you information about helices, sheets and turns. Then you have uh, unintegrated portion. Unintegrated portion means it is recently introduced. The information about the protein is just incorporated. Then it is filtered by the scientists to put them in the respective databases. So here you can see down function prediction is also given, which type of biological process it is involved in, like platelet activation, protein polymerization, blood coagulation, then its molecular function, then its cellular component. And best part is you have to click on this, it will take you to the PubMed article with the geo accession number. Can you see? And PubMed article gives all the information about the meaning also. So even if you don't know what is platelet activation, it will take you to the page where meaning of platelet activation is also given. So it becomes very easy to get all type of information right from the beginning if you are not at all aware of the what kind of the protein it is. Okay, so as then this was about the Interpro scan for function prediction. Now we have another tool which gives you structure prediction and mutation analysis. This Interpro scan, please try it for yourself by giving entry and then click on each and everything. If you click on this database, it will take you to the external link page and there you have to click on the external link and it will take you to the page of that database. So as that is why I told you that it is redirected to many databases. So wherever you click this, this is a screenshot of the result to explain as a demo, as a theory. But when you perform it, actually, it will take you. If you move your mouse, it will take you to the redirections of the entries, and it will give you information or its presence with the accession number in that particular database. Then the next part is structure prediction and mutation analysis by 582. So first part is domain analysis, then is the template analysis, then is investigator, then sequence profile and mutation profile. So how to use this structure prediction and mutation analysis for 582. 582 is also one of the very important and interesting tool to use. It is also very extensive in information. So you can just give the sequence here. You have to give your email ID. Why? Because here this tool has only the limitation. It gives you a lot of information with very interesting graphical display. So the results are also very beautiful and easy to understand. Only the limitation of this 582 is it takes a lot of time because of load on the server. So each result will take at least 20 minutes to run. So we can't afford to lose that time. So that is why the tool has provided us the facility to give email address. So we give this email address and optional job description, then you give the sequence and you click on Fire search. Okay. Now this kind of fantastic result comes. Here you can see secondary structure prediction, which is beautiful green helices. Then you have turns in between, then the confidence interval. Confidence interval meaning always 582 gives you confidence interval. That means how much, what you can say, how well it is giving the accuracy rate. If it is in the bright red bar, then there is interpretation that tool is more than 95% confident about its result. So you get this kind of confidence bar. Then these helix structure, and then here you can see query and model residues. So here you have this other applications also: clashes, rotamers, Ramchandran analysis, confidence disorder. Clashes are the points or the regions where these proteins are clashed over one another. So this region, if you click on clashes, it will give you the color change here. So blue color is always high, good region, okay, and red color region is the clash region. Then you have Ramchandran analysis. Ramchandran plot, you have heard. Ramchandran plot is basically allowable and disallowable regions. So, good region is always in blue color and red is the bad region according to Ramchandran plot, and it depends on phi and psi values. Then you have something which is known as disorder. Confidence I already explained. Then you have something which is known as disorder, and disorder is basically there are some regions in protein which are disordered. That means they do not show either alpha or beta, but a disorder kind of structure. And this disorder region, if they are more in protein, it can lead to malfunctioning of the protein. Now, very important part is here, if you move your cursor on this amino acids, it gives us these two types of profiles. Can you see? Sequence profile and mutation profile. So wherever I'm moving this mouse on the amino acid, I'll focus on one amino acid. So here I have taken this example, F. So if I stop at F, you can see the changes in sequence and mutation profile. Sequence profile, there is only one yellow bar. You always look for yellow bar and red bars. Those are highest or significant values. So if you see here for F, in the yellow bar, F is the region. So that means probability of occurrence of F at that position is the highest. So that particular region cannot be replaced by any other amino acid. So that is the sequence profile. Now, mutation profile is for F, there are certain positions of amino acids which are with, you can see red tall bar and red yellow bar and orange bar. Rest are blue and green that we ignore. So red tallest bar and yellow and orange bars, they indicate that probability of replacement of that particular amino acid is highest, and it is significant also. What do you mean by significant mutation? That means change of amino acid at that particular position will reflect in the change in the either the what you can say properties of the protein or the functioning of the protein. So you see here for F, you can see here G is the tallest bar. So that means at this particular position of A, here the residue number will be highlighted. Suppose if it is 10, 20, so I can say it is approximately 15th or 16th amino acid maybe. So 15th F, if it is replaced by G, since it is showing highest red bar, it is the significant mutation and it will have impact on the functioning of the protein. So it is very, very important from our point of view to know this. If the particular region shows highest significance of the mutation or not. Then below that is this E. So that means if F at that particular position is replaced by E, it is also going to show you high significance of the mutation. Okay. So similar way, orange and yellow. So this also, and blue and green means that particular substitution. That means if it is replaced by V valine, it will not have any impact on the functioning of the protein. So it's that way. So it is very, very important application nowadays to know what mutation will have effect on the protein. So sometimes scientists study this, get the result, and deliberately cause mutation in that particular region, so as to understand how the functioning is changed. Then comes Drug designing. Drug designing is also very, very important part, as I told you. It starts with targets. So why it is important? Because it relieves or it reduces the time. Okay, so that means computational time is basically reduced. Any drug, if you design in wet lab, the uh, time is very much too much involved. Like you design it and to test it. So in drug discovery, which is in silico, you reduce the task, you reduce the energy, reduce the cost involved. That is also very, very important. Now, what is the step? Okay, steps. So first is target identification, then PDB structure, then target validation, simulation, lead identification, lead library, docking, and QSAR is the advanced. So let us see one by one. Okay. So this is like start target identification, target validation, lead discovery, lead optimization, preclinical, preclinical test, and clinical test. Okay. So we start with target identification, then validate the target. Sometimes it is needed to validate the target if the PDV file is not complete. And then we go for lead discovery, lead optimization, and test. So yeah, so then next is simulation. Simulation, why it is needed is because we are developing all this to the virtual system. So it's always ideal to mimic it in the virtual system. So simulation is nothing but study of trajectories of the system of n particles by numerical integration of Newton's equation of motion for specific interatomic potential with initial condition and boundary condition. This is wholly based on molecular interactions, and dynamics play key role in the functionality. So if I have to explain it with the help of this diagram, this is the particle system made up of n particles. These are x, y, z coordinates, and this to each and every particle, I am going to apply molecular dynamics. That means Newton's equation of motion. So for each and every particle, applying Newton's equation of motion will give me at the end trajectory of the system. So studying this trajectory of system gives you better idea of how well the protein is stabilized. So we are stabilizing the protein by running simulation program, and that is NAMD and VMD program. As I'm repeatedly saying, this is all a different together a complete workshop. So in the simulation workshop, we explain how to prepare the input files, how to run the command, how to, after running the command, you will get the stabilized structure, then you analyze the structure. Okay. Then comes protein ligand docking. You must have heard this, all the terminologies you must have come across. Okay. So docking means what? It is like a protein ligand docking we are talking about. There are different types of docking. Main focus is protein ligand docking nowadays is because of this drug designing demand of the drug designing projects, like how well the ligand and drugs act on the protein. Prediction of that is done by docking. So you can see here, for simplicity, I have put this diagram. This is protein and ligand. For a better view, if you zoom it, you can see here, this is the ligand in green color and these are the protein residues. So best way to read this is with the help of docking score. And for that, we use Autodoc Vina tool. There can be different tools, but free is Autodoc Vina, and accuracy is also very high with respect to Autodoc Vina. Now we can see here in the ligand, what is docking basically is it's a fitting like a key and lock or enzyme and receptor. Each structure will perfectly bind to another structure only if there is a lock and key type of fitting, very proper fitting. Now, docking is fitting the ligand on protein in such a way that this particular interaction is energetically and we can say energetically and geometrically favorable. So we get the score in the form of docking score. least is the docking score, or more is least is the binding affinity, we can say, and more is the docking score. You can go for selection of that ligand. So you get 9 modes when you operate it with the help of Autodoc Vina. Autodoc Vina is again a command-based program. It is again a one single workshop of 5 days, which I conduct, and in which I teach all the softwares and programs which are needed for docking. So at the end, you get 9 modes, which we learn to see and visualize with the help of visualization tools like Discovery Studio. Then you have Edit and Primer. So in nowadays, when I take workshop, I teach it with 3 visualization tools. So for your better understanding. So performing Autodoc comprises of first session, then performing analysis is next, and then we perform protein, nucleic acid, and multi-ligand also. So basically, this is the docking, meaning of the docking. Then this is the view of Autodoc. So in Autodoc, you take the protein, you take the ligand, and then you form this grid. So we learn how to read this grid points and mention the scales of the grid box, how to prepare the configuration file. On the basis of grid box, for which our Autodoc tool helps us, you get these values and you put these values and run the configuration or Autodoc. Like this, you get 9 output modes for affinity. As I told you, better is the negative value, highest is the uh, possibility of good docking. So you select first 3 modes ideally, which are crossing value of minus 5.5. Then you have RMSD, which I mentioned. This is nothing but the difference between reference structure and the template structure. Then you have RMSD UP. So on the basis of RMSD, it should be always low. So here you can see for first it is 0, then it is 2, but it is then suddenly increased to 19. So better to go for 2 modes. So these 2 modes are, this is pi mole. These two, all these 9 modes, you can see 1/9, 1/9. So you, all these 9 modes, you can nicely visualize it in pi mole. So this is the surface view of pi mole, and this is the ligand. As you move your cursor or the arrow key on the keyboard towards right, 1 to 9 conformations are reflected in this pocket. So you can see the best one, along with that your energy value. So you select one particular form or conformation which is good with respect to the score also, and which is fitting well into this pocket. This pi mole view is very, very important when you are publishing any results of dockings. Okay. Then last is mathematical modeling or QSR, which is very highly advanced stage of quantitative activity structure relationship. It comes with descriptors, and we have 2D and 3D descriptors. What is mean by 2D and 3D descriptors is the properties, okay, which are 2-dimensional and 3-dimensional. So this properties helps us to give models. Models are built on the basis of the properties. Okay, then mathematical equation is built on this, and most important application is, again, as I said, we reduce the time and cost. So on the basis of this 2D and 3D descriptors, we will construct one model, and which is based or which will be designing mathematical equations. Can you see here? These are some descriptors. So this is the modular structure. These are the properties: 1-dimensional, 2-dimensional, 3-dimensional. These are the descriptors like constitutional, electronic, geometric, hydrophobic. These are, and on the basis of descriptors, VLife and VLife MDS is there, which is a licensed software, and we can perform QSAR. Descriptors will be calculated by the tool, and on the basis of this descriptors, you build a mathematical model. And if you generate this mathematical model, it works vice versa. So if you know the descriptor for unknown ligand, you can get its activity. And if you don't know the descriptor, then from the activity data, you can calculate the descriptor and build the ligand in that way on the basis of chemical structure. Okay, so this is about the QSAR, which is again the one separate module and very advanced technique and very good application of bioinformatics, because it in turn helps wet lab. So this is how topological, geometrical, thermodynamics, electrical, and constitutional descriptors are involved. These are some books which I always advise to refer. Along with that, there are many good books on bioinformatics to start with. Since it is a beginner's core, I always mention it at the end of presentation. You can start reading and get information about what I explained in theory from Mount, Gibas, Bergeron. Then there is Bax-Venice is also, it's not put on the slide, but Bax-Venice is also very good book for bioinformatics to start. And there are many books which you can start as a beginner to understand bioinformatics. There is always fear of this subject in the biotech fraternity. So I always tell in my modules, workshops, and webinars that don't be scared. If you study this particular subject as logically and patiently, and learn this each and every technique step by step, there is nothing interesting like this. And also it is in great demand nowadays, because people in biotech are needed to get expert in bioinformatics in order to provide in silico analysis. So if you learn this, you'll be in high demand with industries also to work on the projects which are in silico. Okay, and it is, you must have realized that in pandemics especially, we are working a lot online. So you can explore for yourself this field, and then uh, you can develop pursue career in this field. Okay, so this is all from my side. I hope all the information was clear, productive, and I have developed some amount of interest of this subject in you. Then also I feel that I am successful in conducting this webinar. Fine. So if you have any doubts, I would like to entertain those doubts and questions. Please feel free to ask in the chat box. And if you don't have any doubt, our team will be putting some links of workshops and courses that we are uh, conducting in this particular field, services which we are offering. Please follow the links, please follow our website and get yourself enrolled in workshop. We will be more than happy to help you pursue your career in this field. Yes, that's all from my side. Please, people, if you want to say anything, if you have doubt about anything. Whatever I have explained, guys, you will be having its hands-on experience in each workshop. Okay, so we'll be happy to make you join there. Okay. Yes, Kabir, you have some question? Yes, please ask. You can type, or if you feel comfortable, you can unmute and ask anything. Better to put it in the chat box in short form so that I understand.
Hello, ma'am. Thank you very much for the lecture. We really appreciate.
Yeah.
So
What,
my
Kabir?
question, I actually studied animal breeding and genetics. So while I was in my final year during my undergraduate studies, we were kind of like, let me say, briefly taught about bioinformatics. And so then, due to the fact that, the kind of situation we find ourselves, so we were not really able to get a lot of things. But when
Yes.
I saw, I was interested to actually participate in it so that I can know what bioinformatics is actually about. You were trying to tell us towards the end of the lecture the opportunities in bioinformatics.
Yes.
So please, actually love to know more about the opportunities like being a bioinformatician.
Okay, so
What
you would like to know about the opportunities you are saying?
yes, ma'am.
Like just now we went through all the applications, opportunities, like you want to work with respect to practical applications or the works you are saying?
Oh, the well,
Job
practical application.
opportunities, job.
Yes, yes.
Job opportunities, like Kabir, today I told you, na, there are many projects which are running on drug discovery, drug designing, and many projects which are run on simulation, also the structure analysis. So these kind of, yes, NGS is also there. So if you can attend the workshops and get yourself expertise in that, and if you get your good hands on docking, NGS simulations, then you can perform or take those tasks as an industry person. You can work on these projects in research labs. There are many labs projects which are being done on this subject, so you can work on those projects. This is all with respect to job perspectives. And also you can proceed or pursue this for your research, like connecting wet lab data, getting a wet lab data and develop it in silico, or take the in silico data first and develop it in wet lab. So there is a huge amount of opportunity if you pursue it properly. This is one line. Then again, it has different aspect which I have not touched also today, which is machine learning and artificial intelligence. In that also you have a lot of opportunities. Apart from that, vaccine design, which is foremost important task right now. Then mutation analysis. So basic and advanced tools itself comprises of so many opportunities and tasks that you can do. Apart from that, you have drug discovery also, as I mentioned. So docking, QSAR, mathematical modeling also. You have many projects where you have to develop mathematical models. So for that also you learn different tools. So depending upon what tool you are using, there are different job opportunities. You have to explore, you have to apply in the labs for different kind of projects they are involved in. Then you can get stipend also depending upon the project. Then you can pursue your career for PhD also in that topic. Okay,
Okay, ma'am. Thank you very much. I really
most welcome.
appreciate.
Thanks a lot. My pleasure. Kabir, you can connect with me from the BDG platform. You can get my email address, or you can put anything which you require to our team. You can mail it to us. My team will connect to me.
Okay, ma'am.
Okay, Kabir, all the best.
All right, thank you very much.
Yeah. You're from which country?
I'm from Nigeria.
Nigeria.
Yeah, thank you very much.
Thank you. Thank you. So, with the team's consent, shall I end the meeting? Yes, team. Okay. Thank you so much.
Yeah, you're welcome, ma'am.
Yeah. So I'll end the
Thank you, ma'am.
Thank you. Yeah. We'll end the meeting now.